Now you see here we are beginning with the post-fertilization event. The post-fertilization event begins exactly after the fertilization. So in our previous class, when we were dealing with fertilization, I said that in flowering plants, double fertilization happens. So this double fertilization, I said again that it is a very, very unique property of a flowering plant where double fertilization happens. That means what? Here we get triploid cells and a diploid cells. Okay. Now you see the central cell that was known as the I said that these three PEN. See, primary endospermic nucleus. So these primary endospermic nucleus, they are triple fertilized. They are 3N, and whereas see these two, actually see, this is the antipodal end and this is the chalazial end. After fertilization, the embryo gives a turn. Okay. Now earlier this was here and this was here. Okay, so the total axis gets turned up after fertilization. Okay, and this is not in your syllabus, so I will not go into the detail. So this particular part, it is actually the antipodal cell that was here and it has come down here, and ultimately this antipodal cell completely degenerates. Okay, and the central cell, okay, that moves a little upward, and here the synergid cell, that is known as PEN, that is primary endoplasmic nucleus, it is here, and we have the 2N embryo here. Now later on, this primary endospermic nucleus, they completely covers up the embryo rather, and here we have got a single mass of cell here. Okay, now if you observe clearly, this is actually the zygote. Okay, and this is covered by, the entire thing is covered by PEN. That means PEN randomly divides by the process of mitosis here, and it completely cover up the nucleus. Okay, now here and this position, you see this, what we see is actually the endospermic cells. Okay, endosperm. And what is endosperm? Now endosperm is actually having the reserve food and provide nourishment. See, what is PEN? It consists of the reserve food to provide nourishment to the growing embryo. Okay, now you see this one has to divide into two. That means the 2N, that is embryo, that divides into two. It is actually the embryo here, and the second nucleus you find here, actually they starts, see, the division is on the horizontal plane. Okay, so this horizontal plane division, it creates a suspension. That means a small tube like structure, that is called the suspension, and here you see a mass of cell, that is known as a globular mass, develops down here. So this is actually a stage where the embryo is developing into a seed. Okay, now you see this mass that is in the globular form, here it takes a heart shaped structure. Okay, now since this heart shaped structure, again if you see here, it gets a turn, like this type of turn here, and you see this and these two type of these elongates. What appears primarily as a heart shaped structure elongates like this to a leaf like structure, and this bends. Okay, so when this bends, these are two portions. This is the radical and this is the plumal. Okay, now once the radical is there, okay, now what I was talking of, you see here what appears as a heart shaped cell, it comes here like that and it develops a leaf like structure. Okay, now this part forms the, you see it is written as a radical and this part is known as a plumal. Now if we consider it to be in detail, you see this radical, okay, see R stands for radical and this is the plumal, and this actually gets a turn. Okay, I'll show the diagram that would, now it appears straight to make you understand, but here it is this type structure. Now once it happens like this, this plumal gives rise to the shoot structure, and this radicle goes down and it forms the root structure. Now this radicle in plumule is completely an dicot thing. Okay. Now see, now till now we were dealing with the endosperm development. Okay. Now here you see we have the embryo development. What happened in the embryo development? That cell divides into a mass of cell. Okay. Just this cell develops into a mass of cell, and we call it as a proembryo. What we call it? It is a proembryo, and you know this development starts from an axis. Okay, for example, you see it, this is the axis. That means the straight structure is here. Now if we consider it, see, for example, if you have seen a gram cell developing, see how I am drawing it. That means what? This is the cotyledon. This cotyledon actually develops from the endosperm, and you know in some dicot plant this cotyledon completely gets abolished with the development. If you see, you can soak a few gram seeds, and if you see the germinating procedure, this cotyledon completely goes away because this has the stored food for these. Okay, so this is the root-like structure. Root-like structure. Now this root actually develops from the radicals. Okay, this develops from the radical, and here you know the leaf-like structure, it is the plumule. Okay, plumule, this is the first shoot, first leaf. Okay, this is the first leaf-like structure. You can see here on the board. Okay, so this first leaf-like structure, it comes out from the plumule. That means plumule has got cells that initiates the shoot system. Okay, and radicle has cells that initiates the root system. Basically these if we consider it as tissue wise, the radicle and the plumule both have got masses of meristematic tissues. You know what are meristematic tissues? Meristematic tissues are those tissues that is capable of actively dividing, and so that this radicle elongates, okay, into a root-like structure, and the shoot, see this cotyledons has stored food. Why? Because during this part of the development, until the seed germinates, it cannot prepare its own food. See, once the first leaf structure develops, these first leaf structures, they are capable of producing their own food, preparing their own food. That means until and unless the green leaf gets mature, they cannot prepare their own food. Okay. Now, if we consider the again same axis like structure we have to draw, and then for example we draw like this. Okay. Then we have here like this. So this is a maize seed, you know, maize seed, and you see this is known as scutellum, scutellum, scutellum. Now what is this scutellum? Scutellum is like that of the plumule, and this is supposed to be the first shoot initiator in case of monocot. Monocot. Okay. Now you see here, these are all endosperm here. Okay, but instead of radicle and plumule, we have, see, we have the, see, coleoptile and coleorhiza. Coleoptile, and here this is the coleorhiza. Now what is coleorhiza? Coleorhiza again here, they are root initiator. What does it initiate? It initiates the root. And what is coleoptile? They initiate the shoot system. Clear? You can see here the same thing, you know, and this is the scutellum. And so this, and you see this is the axis of development, and on the basis of axis of development, germination has been termed as, see, epicotyle, hypocotyle, like that. So for example, if the seed is here, it develops this hump-like structure. For example, like this, it comes out, okay, like this. So it is epicotyle. Once it is within the soil, it is hypocotyle. Okay, so this will come to later on when we'll be dealing with germination. Here you see this diagram that I was dealing on the board. You see, you have globular embryo, you have suspension like that, okay, radical, see cotyledons are there, plumal, and here again what I was telling you, see this was this I was telling you that it gets a turn. See, this I was telling like this was drawn straight, but literally it gets a turn like that. So it is turned and condensed like that. Okay, see the radical is here. Okay, epicotyle, then hypogeal germination, that means it comes out like this. Okay, and here you see this is the scutulum. Okay, you have the coleoptile is equivalent to the plumal, and here you have the coleorhiza. Okay, root apex and shoot apex is like that. So go through it properly.